about the mechanism of DNA replication. Now, before we talk about the mechanism of DNA replication, let's talk about where and when DNA replication happen. If you remember, this is the cell cycle. These are the phases through which a cell goes, going from one cell. It goes through the phases of G1, S, and G2, and then during the mitotic phase, it splits into two. So before a cell can divide, first it has to replicate its DNA. It has to make a copy of its DNA. And when does DNA replication happen? It happens during the S phase of the cell cycle. So if you remember, the DNA is an anti-parallel double helix. And the way the two strands are held together is by hydrogen bonding between the nitrogenous bases that are sticking out of the backbone. Now, how does the cell know how to make an exact copy of its DNA? Well, it uses the fact that these nitrogenous bases are complementary. So if you were to separate these two strands from one another, then each of these strands can act as a template to form a new DNA strand. And how does the cell know how to make the other strand? Well, it knows that a nucleotide with a thymine nitrogenous base hydrogen bonds with a nucleotide with an adenine base. So then it knows to bring a thymine here, and the same pattern continues. The mechanism of DNA replication is said to be semi-conservative. And the reason it's semi-conservative is because when you start with what's called parent DNA molecule, so these are the two strands, we're not showing them as the double helix just by straight lines, just for simplicity. So the gray molecules are the two strands of a DNA that you start with. But when this DNA undergoes replication, the two new DNA molecules, the two new double-stranded molecules that you end up with, are composed of one strand from the old DNA and one strand from the new DNA. So that's what it means when we say DNA replication is semi-conservative. It's not, if it was totally conservative, then you would get one double-stranded DNA that had two blue strands and one double-stranded DNA that would have two gray strands. No, that's not what happens. So what you get, the two new DNA molecules contain one strand of new and one strand of old. Okay. So before we can talk about DNA replication, there is one feature of nucleic acid polymerization that is very, very, very important to understand. And the point I want to get across is for you to understand that nucleotides, when they polymerize, they are only added to the 3' hydroxyl end of another nucleotide, not to the 5' phosphate end. So making a polynucleotide always requires a free 3' hydroxyl. Otherwise, you cannot make a new DNA. So what does that mean? So this is a nucleotide that is sitting here. It's a monomer, and you want to make a polymer out of it. How does that work? Well, you bring another nucleotide, and then through a reaction which you've heard of, it's dehydration synthesis reaction. You remove a hydroxyl from one molecule and a hydrogen from another molecule. You make water, and then as a result of this, you're going to make a covalent bond between the two nucleotides. This red line is supposed to represent a covalent bond. Okay? So as you build the polynucleotide, you're going to bring the next nucleotide. And you notice that this addition of the nucleotide, this new nucleotide always comes to this end of the growing strand. It's not added to the phosphate end. So the process continues. This is what happens in nature. What does not happen is in nature is what I'm going to show you right now. So here's a nucleotide in the bottom. And then when you try to polymerize the DNA, make a polymer of the nucleic acid, you'd bring another nucleotide, but then you would add the new nucleotide to the 5' phosphate. Okay, but this does not happen. This does not happen in nature. Does not happen in nature. So therefore, a nucleic acid, whether it's RNA or DNA, never grows from the 5' phosphate end. You always need a free 3' hydroxyl that's already there, to which you can bring in a nucleotide and add to the growing DNA. Very important to understand and remember this. I wanted to take you through the steps of DNA replication and actually draw it by hand. So as we know, the DNA, I'm going to do my best to try to do nice drawing. I'm not very good at drawing. So let's say we know this is a double helix. So this is a double helix. Let's say this, the top strand is 5' to 3', and this one, as we know, goes the opposite direction. Now, DNA replication doesn't happen everywhere on a DNA molecule. There are specific regions on the DNA molecule which we call origins of replication. That's where the replication happens. So let's say if we have an origin of replication here, it's a specific sequence, and maybe we have another one here. So what happens is that the DNA actually unwinds around those sequences, and then we have something like this. Okay, so when the DNA unwinds in order to be replicated, you can imagine that if you had a sort of a string like the one shown on the top, if you open it up, and then you're going to create basically, let me bring my laser, bring my pointer, then what you can see is that around areas outside of the place where the DNA opens, you're going to create tension on the two sides, and that's not sustainable. So therefore, what the cell does is that it employs an enzyme called topoisomerase, which is going to come in, and I'm going to draw a topoisomerase with red. So it's going to come in and sit on the outside areas where the DNA is kind of being super wound, and it's going to unwound these regions so they're not so stressed. So I'm going to kind of draw it up here and say this is topoisomerase. Okay, so then how does this DNA open up in the first place? Well, it opens up using another enzyme called helicase. So then it comes in and it opens up the DNA molecule. Okay, and then what happens is that these origins of replication, as DNA replicate itself, it gets bigger and bigger, and then these two origins of growing regions of DNA, which are growing, this one and this one, they're going to join, and you're going to get replication completed. This molecule that we show like this, the helicase. So it opens up the DNA. Now we know that strands of DNA, opposite strands of DNA, do hydrogen bonding. So if you are going to open up this DNA, then it's going to snap back. So what then? So the cell utilizes proteins called single-stranded binding proteins, which I'm showing by these green little dots. We show it as SSB, which holds the DNA molecule far apart from one another, so then the enzyme that copies its DNA called DNA polymerase can come in. The DNA is opened up, it's kept open using the SSBs, and then topoisomerase is relieving stress. Now you have to make copies of the DNA. And if you remember, we said that DNA only grows in one direction. 5' to 3'. And you also remember that for a nucleotide to grow, we need a 3' hydroxyl available, so then a new nucleotide can be coming in and added to it. So where does the DNA polymerase, the enzyme that does the polymerization, where is it going to get the 3' hydroxyl from? Well, and enzyme called primase, called DNA primase. I'm not going to draw it in here, just imagine that it's there. What it's going to come is going to come and make short RNA fragments which have 3' hydroxyl available. Okay, so it makes 3' hydroxyl available, and this is actually an RNA sequence. So this line is an RNA primer. So for DNA to be able to replicate itself, it depends on a short piece of RNA, and both strands these RNA pieces are created, and then the DNA can be polymerized in this way, and in the opposite strand it would go the other way. Okay, and which molecule will make the copying? It's going to be a molecule of DNA polymerase. So this DNA polymerase is going to make a DNA copy of the strands that are opened up. Now, I want to draw your attention to and focus on only one part of what's going on here, and I'm going to show you. Let's see, let's highlight it with this pen. This region that is being polymerized. You see these two strands are opening up. Helicase is opening it up, and it's growing. This is called a replication fork. So let me draw it again. That's just too busy. So if we have this thing, okay, so this area that is a growing tip of the DNA, where it is being opened up by this helicase, that is called a replication fork. Okay, so let's take a look at what is happening in the replication fork. I'm going to blow it up in here. So right now, I'm just going to forget about these areas, all of it, and I'm just going to focus on this area. So let's imagine that this, we're just looking at this. That's the replication fork that is going outside. And we're not going to worry about the proteins and the helicase and the polymerase or anything like that. We're just going to worry about the DNA that is being made. So we're also going to need the RNA primer. So if the top strand goes from 5' to 3', the opposite strand of it should be anti-parallel. So here's the RNA primer, 5' to 3'. So then what happens, the DNA polymerase comes in and then extends this DNA. And it keeps going and going and going and makes one large piece of DNA. Okay, so this is the top, this one that I just drew. It grows in one piece. So it grows in one piece. Okay, it grows in one piece. Now, what do we call this strand that grows in one piece? We call it the leading strand. So note that the leading strand is the strand that is being newly synthesized. It's new DNA. Now let's take a look at the opposite strand. So if the template that we're using is going from 3' to 5', then the one that is being synthesized has to go anti-parallel. So it would have to go in the opposite direction. Except in the other strand, you're not going to continue building this in one piece. What happens is that the other strand is going to be synthesized in pieces. So then the other strand is being synthesized in pieces, in fragments. Okay, so this strand that is being synthesized in pieces, let me show it to you using my arrow. So this strand, this new one that is being synthesized is in pieces. We call it the lagging strand. Why do we call it in lagging strand? Well, because it lags behind the leading strand, because it's made in little pieces, and those little pieces have to be connected with one another. And how are these little pieces connected to one another? Well, two things need to happen. So we're going to remove these primers. DNA polymerase is going to build whatever we've removed, and then the fragments have to be joined together using DNA ligase, which is another enzyme that is involved. Now these fragments in the lagging strand are called the Okazaki fragments, after the scientists that discovered it. So DNA replication happens on the two strands. One of the strands is built continuously, which we call the leading strand, this one. The other strand is being synthesized in fragments, in little pieces, which we called Okazaki fragments. So then at the end, you're going to remove, as we said, remove these RNA primers, and then DNA polymerase is going to fill in the gap, and then DNA ligase is going to join them. Ultimately, also at the end, the cell is going to remove these RNA primers also from the other strand as well. So that is a detailed summary of DNA replication. Okay, so you saw a detailed drawing that I tried to do, show you the detailed mechanism of DNA replication, and this is a more simplified version of it, except we don't have all those other proteins involved. So we have the replication fork in here. That's the replication fork. Another one is here. And then you see on the top, this, if you were to look at this end of it, here's the leading strand, this one. This one is the leading strand. And these are the Okazaki fragments. Okay? So now, if you take a look at this DNA, an interesting problem arises for this cell. So if we were going to extend this replication all the way to the end of the molecule, we would have this green primer, RNA primer, okay? And then we would have the red DNA that is being made. And mine doesn't look as pretty as the ones you have here, okay? And we said that the cell is going to remove the RNA primers. So it's going to chop this off, right? It's going to chop it off and remove it. Now, in the middle, when it chops off this green stuff, DNA polymerase is going to come and fill in the gap in here. But that doesn't happen at the end. There is no filling in the gap on this end, and also on the other end. It's here. So what happens at every time DNA replicates, the two ends of the DNA become uneven and one strand becomes shorter. Just like in here, that is shown in here. So here it is, the DNA that has been replicated, but on this end, you come short because the cell removes that RNA primer and doesn't replace it with anything else. Now, there is an enzyme that was discovered by this scientist, Elizabeth Blackburn, and she won a Nobel Prize for this discovery. And the enzyme is called telomerase. And the job of the telomerase is to come in and fill in these ends. Now, the ends of our DNA molecules have repetitive sequences that are very characteristic to the ends of the DNA molecules. And that end sequence is complementary to an RNA that is part of telomerase. So telomerase comes in and fills in these ends that have been left hanging. So using the RNA that is part of the telomerase and by complementing to the template strand that is hanging, telomerase synthesizes, builds the end of the DNA. So it rebuilds the part of the DNA that is chopped off. So once this end becomes long enough, then that allows the DNA polymerase to come and filling the gap. That's the job of this enzyme telomerase. It rescues the ends of our chromosomes from becoming ever so shorter every time the DNA is replicated. So here it is, a true or false. These are all number one, but that's okay. The true or false questions, there are 4 true or false questions. There's one strand of DNA, and just make a complementary strand, and don't forget to put these notations of 5' and 3'. And next, replicate this piece of DNA. So a good way for you to practice this was to maybe put the first G and the bottom C here as the beginning of a replication fork, and then build the rest of them on the DNA that is open. So this would be 5', and that would be 3'. And then write the rest of this sequence, the top one, write it on the top strand here, write it in here, and then the bottom one, and the bottom one, write it on the bottom strand, and see if you can recreate a replication fork. Don't forget the Okazaki fragments, don't forget the RNA primers.